Alright, so this video is for how we see, like the physiology behind it. We've talked about the anatomy, now we're going to talk about how vision is actually, how it works. So, refraction of light is a bending of light. So the ability to see something, it has to be projected in the back of the eye on the retina. And light will bend if it goes into different density media, different densities of air, of solutions, of lenses, so it'll bend. So as the light goes through the eye, as it goes through the cornea, and then the fluid, the back of the cornea, the aqueous humor, as it goes through the lens and it goes through the vitreous humor, it's going to bend, or in different ways. And those, that's called refraction. So they bend as they go towards the retina. And so a refractive index, each one of those media, it could be a water, it could be air, it has a refractive index, or the amount that it will bend something. If you go ahead and if you put in a glass of water, put a spoon in it, a spoon is going to look different from how it bends the light looking through the glass. So when you're looking at something that's close to your eyes, a few things are going to be different than looking at something that's far away. So when you get close to something less than 20 feet, that's called the near response. These three things are going to happen. Your eyes are going to converge. That means the eyeballs will both move medially. The lens is going to accommodate. That means get thicker. And the pupil itself is going to get smaller. It's going to constrict. So the first is convergence of eyes. The extrinsic eye muscles pull the eyes inward. This is if it's really close, if the object is really close, you're going to look cross-eyed. And the idea is to refract the image even more because you're so close to it. You need to bend the light even more. And if the eye muscles are weak, it's really hard to converge. So somebody that has a lazy eye, their eye has trouble doing this because the muscles around it are weaker. Accommodation of the lens, that's just a fancy word for the thickening of the lens. Thickens the lens. So what happens is the muscles that hold the lens, remember those are the ciliary muscles, they contract to thicken the lens, and the suspensory ligaments relax, and so the accommodation means the lens thickens to increase the refraction or increase the bending of the light. Another thing, like I said before, your pupil will constrict. The inner layer of muscles called the sphincter muscles in the iris, they will contract to make that lens smaller, or the pupil smaller, to direct the light specifically to one area in the lens. Okay, so that's when you're looking at something close. When you're looking at something far away, it's different. Your eyes face forward, okay? The lens, instead of getting thicker, it's going to become flatter, and the ligaments are going to be pulled taut or tight. And the pupil is going to be larger to allow more light, greater amount of light into the eye. Okay. I should also mention there's a good picture or a good figure in your book that goes over those two things, what happens when you're looking at something close or when you're looking at something far away. I know on page 624 there's a good image that talks about corrective lenses and how they help if you're nearsighted or farsighted, in page 621. Also, I should mention that when you look at something, when it's received at the retina, it's actually upside down, and then your brain fixes it. So the retina receives everything that I'm looking at right now in Caribou, it receives it upside down. And then once it's sent to the brain, your brain flips it back to the right side, which is kind of cool. Okay, so Next, we're going to talk about now the light is at the retina. Now what happens? Well, light is a form of energy and it needs to be converted into electrical energy and for your brain to be able to interpret that. And that is performed by a type of receptor called a photoreceptor. And we'll talk about the specific types. These are your cones and your rods. And they're on the photoreceptors, the rods and cones are on the outer segmented layer of the retina. And they're carried in these discs and they were placed regularly. Now, you have about 100 million rods per eye and you have 10 million cones per eye. So rods are longer and narrower in shape and they're more numerous, like I just said. Quite more numerous. Each rod is sensitive to low energy light, dim light. They're very sensitive to dim light. They're not active in bright light. Your peripheral retina, so on the sides and then the insides, contains a lot more rods than the center. Okay, so rods will pick up on the light energy and they'll send it to the next cell called a bipolar cell. Many rods, so let's say this is 5 rods, many rods converge with one bipolar cell. So all 5 of these rods are going to tell this one bipolar cell what it has seen in the dim light. And then that bipolar cell will then pass the message to a ganglion cell that sends it on the optic nerve to go to the brain. Okay? So many rods converge on one bipolar cell. Now this has a negative point to that. Because of many rods converging on one bipolar cell, the image is slightly blurry, but it can pick up on a lot of information. Okay, so peripheral vision, you can see a lot of things, and in dim light, you can see a lot of things, but they're blurry. Cones, they're a little bit different. So they're not in the periphery; they're highly concentrated in an area of the eye, the back of the retina, called the fovea centralis, which is in the macula lutea. Okay, there's many cones highly localized in that area to produce a very sharp image, and they're activated by high intensity light, and they allow color vision, cones, high intensity light, daytime. They have a 1-to-1 relationship with bipolar cells. So remember my analogy with my hand. This is one cone, one bipolar cell. One cone will tell one bipolar cell what it saw. There's a 1-to-1 relationship. Just like in our school, every student has one computer. Okay, teachers have a computer. Cones have a 1-to-1 relationship. So because of this, they can produce a very sharp image in bright light. We can see it clearly. And this just shows in the anatomy, again, review here. Here is the retina, and these are the 3 layers. The bipolar, or the photoreceptors, the rods and cones are in the back of the retina. They will send, so this is a cone, these are the rods. The light is coming this way, hits the back of the retina, and these will pick up on that light energy and convert it into chemical or electrical. They will send it to these are the bipolar cells. The bipolar cells will send the information to the ganglion. So, photopigments are in the cones and rods, the photopigments. Photopigments are made of a protein called opsin and a light-absorbing part called retinol. And retinol is made from vitamin A. This is why vitamin A is important for eye health. Different pigment types are because there's different opsins, and they can each detect a different color of light. So rods all contain rhodopsin pigments. Cones, there's 3 different types of cones, and each one contains its own pigment, its own rhodopsin. Some cones detect blue light, which are short wavelengths. Some cones detect green light, which is intermediate wavelength of light. And some cones detect red light, which are long wavelengths. Okay? And this is a graph that just shows the different wavelengths of light that exist and how blue is a short wavelength and red is a larger wavelength. If you're colorblind, you have, well, first of all, it's genetically X-linked, meaning it's carried on the X chromosome. And for that reason, it's more common in males, because males only have one X chromosome. So that if they inherit color blindness on an allele, on the X chromosome, they're more likely to show that disease because they don't have another X to protect themselves. To be colorblind, you need one cone cell to be absent or deficient. And there's 3 different genes or alleles for genes for color blindness. And you can be absent in all of them, or one of them, or a few of them. So there's different types of color blindness. The most common type is red and green color blindness. And that just means they have difficulty distinguishing whether something is red or green. And whenever I think of that, I think of stoplights and how they must see the stoplight colors and be totally different than us that aren't colorblind. Phototransduction is a fancy word for the light energy being converted to electrical energy. So I'm only going to talk about how this happens in the rods or in rhodopsin, but it also does happen in the cones. Okay, so in the dark, rhodopsin contains a certain form of retinol, and it's called cis-retinol, and it's kinked. When you go into the light, what actually happens in the rhodopsin is that retinol changes shape. It goes from kinked to straight, and that is called transretinol. And this is termed bleaching. It will then leave the opsin. Okay? After that bleaching, meaning it's not dark any longer, it's not a bluish color, it's white essentially, it needs to be rebuilt for you to be able to see in dim light again. So this figure shows the bleaching reaction that happens in rods. So in dark, the opsin is there, that's that protein with the cis-retinol, that kinked version. When light comes into the eye, it causes essentially it to be non-functional, rhodopsin to be non-functional. And that's why it hurts a little bit when you go from dark to light. It's because this cis-retinol is changing shape and leaving the opsin as you see there. All right, boom, it leaves. And then when you go back into the dark, it needs to be rebuilt, this rhodopsin. So this is the reverse reaction, and you need energy to do that, how it goes from straight back to curve. Now, moving from the light into the dark, from the dark into the light, you know that things are happening with your eyes. You know that your eyes have to adjust to those situations, and those situations have names. Dark adaptation is when you go from the light into the dark. So it's return of sensitivity of your eyes after going in the bright light. You have to be accustomed to the dark again. So remember, you're in the light, you're enjoying the outside, the sun. You go into a very dark, maybe a movie theater or something like that. All the rods, remember, are bleached. Okay, they're all straight. They need to be rebuilt. Okay, for your eyes to get used to the dark, it takes about 20 to 30 minutes for all that rhodopsin to be built up optimally. And then you can see well in dim light. The opposite, when you go from a dark environment, from out of a movie theater,